Hey guys, it's Mrs. Frazier. This is How to Do Science Part 1, and in this video we are going to talk about the process of science and how to write our hypothesis. The scientific method is a way that scientists think about figuring out a problem. You've probably seen it in your past classes, and it has looked something like a series of steps, like 1, make an observation, 2, come up with a question. Well, the scientific method is not a list of steps that you follow like a recipe. It is actually just a thought process. It is a way to approach a problem and come to an accurate, evidence-based conclusion. You already use it in your everyday life without knowing it. Let's take, for example, you are making cookies. You're following a recipe, but the recipe does not tell you how long to put them in the oven. So your question is, how long do I cook them for? You formulate a hypothesis based on some previous observations, like you kind of remember maybe 30 minutes, maybe 20, somewhere around there. So your hypothesis will be, I'm going to put this batter into the oven for this amount of time. And then you come up with some testable predictions. So you're going to make 3 different batches using the same batter on different pans, and you're going to put them in the same oven for different amounts of time and see which ones come out the best. You put them in the oven and you gather your data. You gather your data. The 30 minutes was too long. They're burnt. 20 minutes too short, and so on. And then from there, you can refine your hypothesis. Like, actually, I'm going to use 19 minutes instead. And then you do it again until you come up with a perfect time and your cookies come out perfect and you figured out a theory for how long it takes to make cookies. Observations lead to questions. Just like in our previous example, observations that you had taken from previously cooking led you to think about how long the cook time for the cookies will be. In AP Bio, when we are doing experimentation, observations are going to come from what you have been learning and experiencing and other experiences that you have had in labs and activities. You're going to use those observations to come up with questions. Scientific questions are experimental. They are testable. So when we are learning things in science, we might be learning these big, broad ideas, and you need to be able to apply them to different situations. So you will take your observations from what we are learning and apply it to a situation where you're asking new and different questions. There is a difference in questions when we're talking about experimentation. There are scientific questions and there are non-scientific questions. Non-scientific questions are broad, they can't be tested, they're easily answered with a yes or a no. It's something you already know the answer to, or they're very vague. For example, if you asked, will these plants grow? Well, of course we know that plants are going to grow. So the answer is kind of like probably, yeah. I mean, is that testable? Yeah, we can test it to see if those plants will grow, but this is not actually a scientific question. A scientific question needs to be testable. It might have a complex answer that needs to be explained. It might be a question that has to be broken down into more than one experiment. It is specific to exactly what factors you're wanting to test. It's built on your knowledge that your scientific knowledge that you're building. It asks what would happen if, and it helps solve a problem. So if we are going to revise this, will these plants grow? Well, maybe we are going to test, will the type of light increase plant growth? That is a more testable scientific question. You can change this out with light. Will the amount of water, will the type of plant grow differently in different places. So you keep it specific. And yes, we're still going to be able to answer this. Will these plants grow? But we're going to build specific pieces of evidence with these specific questions that we can put together to answer a bigger question. Coming up with a good testable scientific question is not just about coming up with what pops into your head first. They are carefully planned. They are based on observation. So in this example, you see that there's 3 different observations, and it comes to an idea that can be turned into a question. In this idea of reasoning and logic, this is a general premise, meaning that this idea here is something that you have been taught or is a common knowledge fact, but then you are asking a question to apply it to a specific and different situation. When you are designing an experiment, you need to think carefully about the variables you're going to use in your experiment. A variable is a word that means a quantity or condition that can change. This word, you've probably heard it in math. It's a common word. It's not a science word. A variable, for example, would be like, what are the variables that are going to help me win this game? Maybe if I practice more, maybe if I lift some weights. That is a common way of using the word variable. And in science, it's the same way, but there are specific variables we talk about within an experiment. We have the independent variable, which is a variable that you can control. And I want to be careful with this definition here because sometimes if it's an experiment that's not being done inside of a laboratory, it's more observational. You can't really control that experiment, but it is the one that you are looking at. When this changes, what happens? So this would be the variable that is the cause, and then the dependent variable is the effect. The dependent variable is a variable that you can observe and measure. So because of this change in the independent variable, what does that do to this variable? That's what you're going to measure. And then this controlled variable, which is also called constants, which I like to use the word constants because control gets so confusing because look, we've already seen it in this definition above. But anyways, a controlled variable means what variables are going to be kept the same if the other 2 are changing. And the reason for a controlled variable, making sure we know, is because if you are testing, like up here, let's look at our example up here. Does the type of light change the growth of plants? So we are looking at light and growth of plants. So your independent variable is the thing that you are going to change. So if we're testing light, we might want to test green versus white light. So our independent variable would be color of light. And then what is the effect of that? That's our dependent variable. We want to see how that changes the growth of plants. So maybe what are we going to measure? The height of plants. And if that's what we're testing, then our controlled variables, everything else has to be the same. So the type of soil needs to stay the same, how much you water them, the location of them, the weather, the climate, everything else has to be the same. And why is that? Because if you change, let's say the amounts of water and you're watering one plant way more than the other, then how do you know that the color is connected to the height of the plants? Or maybe it was that difference in the amount of water that changed the height of the plants. That's why you have to keep the variables that you are not testing controlled the same for all parts of the experiment. Let's do one more example. Does leaf size differ in populations growing in shade versus full sun? What's our independent variable? It is the thing that we are changing. So we are changing shade versus sun. So we are changing the amount of light. And depending on that amount of light, what is going to change? We want to know if leaf size is going to change. So that's what we're going to measure. What will we have to keep controlled in this experiment? It would have to be the same type of plant if we're measuring leaves. Our measurement type, how we are measuring the leaves, has to be the same. What else? Location, as in like climate and weather, and so on. We touched upon this previously. There are different types of experiments that we can do. There can be a controlled experiment or an observational type of experiment. Controlled happens most often in labs, and that is because the scientist has full control of all of the variables. Researchers are able to determine which of their subjects receive the factor that is being tested for having a causal influence upon another factor, aka they can control the independent variable and measure the dependent variable. This example over here is a controlled experiment. As you can see, the scientist was able to set it up. They're able to add water only to one and not the other, and then they are measuring the two different groups compared to each other. An observational study, remember, science is nature. There's things that we just can only observe. We can't test on them because it's either illegal or it will hurt something or whatever, or it's too big or too expensive. Observational experiments are when researchers have no such control over whether their subjects receive the treatment being investigated, but they observe, they take notes, and they come to conclusions based off what they're observing. For example, if a scientist wanted to study the height of the trees in this forest, they are not going to be able to change the weather, the sun, the rain, the location, or the types of trees. What's there is there. They will just be able to observe what is going on and take all of that information, put it together to come up with a logical conclusion. Before beginning your experiment, you need to write a hypothesis. And a hypothesis is just a prediction of what your experiment is testing. What do you think is going to happen? And then what happens after you do your experiment is you either accept your hypothesis as supported, or you reject your hypothesis as not supported for your experiment. And what does that lead to? That leads to probably a new question or a different hypothesis. It doesn't mean that that study is over. It's just the next step. So we do two types of hypotheses in AP Bio: a research hypothesis, and you will also see a null hypothesis. Another word for a research hypothesis, it's also called an alternative hypothesis or an experimental hypothesis. And you are most familiar with these. These are in the if, then, because format. And this is where you take your variables. If I change this, this is always your independent variable, then this will change, your dependent variable, because, and this is based off of your science research that you're using to come up with this scientific question. If we take this question up here, does leaf size differ in plant populations growing in the shade versus sun? How do we turn that into a research hypothesis? If a plant is grown in shade, then it will have larger leaves because, and now this because part is not just a guess. This is going to be based off of what we are studying, and this is where you apply the theories that you're learning in your textbook and our activities, and you apply it to a different situation. So this is kind of where the science, well, like, why are you thinking this is going to happen? And I know we're not studying this, but we know that sun is required for photosynthesis, and photosynthesis is what helps plants grow. So if it's in the shade, it's not getting as much sun. So maybe that's why it needs bigger leaves, so it has more surface area to get the sun that it does get. So your because would be because plants need light to grow, and shady plants will adapt to have bigger leaves to absorb more sun, something like that. The other hypothesis that you will be asked to write, and sometimes you're asked to do it together, but most of the time it's one or the other. And the reason for the null hypothesis is to do some stats. It's really there for when we have to do certain statistics. And the null hypothesis is very confusing for many students, but it's actually way, way easier than the research hypothesis. All it is saying that there is a zero effect due to the independent variable. And then if the null hypothesis is true, the difference in the dependent measure are due solely to chance fluctuations. Let me sum this up for you. The null hypothesis, all it's saying is there will be no statistically significant difference between the experimental group and the control group. And the way that you write a null hypothesis is the same no matter what. You don't have to do the background research. I mean, you still do, but when you're writing your null, you don't have to include it. It's just saying there's not going to be a difference. And if we do see a difference, we're saying that we think that that difference is only going to be due to chance and not actually something scientific going on. So if you look at this figure up here, you can kind of see that the research hypothesis is connecting the independent variable cause and effect. But when you write a null hypothesis, you're stating that there is not going to be this independent variable is not going to affect the dependent variable. Again, those are just two different types of hypotheses you'll be asked to write. Let's try to write it for this experiment. Our research hypothesis, so it looks like here we have some water. It says plus water minus water, so we're going to go with that being the experiment. Our research hypothesis would be if water is added to a plant or to, let's say, seeds, then those seeds will sprout because, and I'm not going to write it all, but because plants need water for photosynthesis. If we're doing the same exact experiment, but we are asked to write a null hypothesis, the way you write the null hypothesis is if, oh, no, it's not if. There will be no statistically significant difference between the experimental group and the control group. Let's talk about these different groups of the experiment. We have control groups, and this is when you're designing and setting up an experiment, and you also have an experimental group. Let's start with the experimental group. This is the group that is receiving the treatment. So if we go to the example previously, it would be the group that's receiving the water. There can be more than one type of, or one experimental group. For example, in our cookie, we would have the different cook times would be different experimental groups with that changing independent variable. The control group is the group that's usually not receiving treatment it's under normal conditions or it's not getting anything. In medicine, it would be called the placebo group. And the purpose of the control group is for comparison. And the reason for this comparison is we need to know that whatever's happening in this experimental group, we need to be able to compare that with what is not happening in the control group so that we know that what is happening here is because of our experiment, because of our independent variable, and not by the environment or just normal behavior. So when we talk about control groups, it's either the one not receiving treatment, but there are also things called positive and negative control groups. And if you are not taking notes, I would take notes on this part. A positive control is an experimental control that gives a positive result at the end of the experiment. As the scientist, you make this positive control and you do it in a way that you know that whatever you're doing is going to have a positive result. It gives the positive result. It responds to the experiment the way that your hypothesis is saying, and it is ensuring that your test, it is possible for it to work and nothing's broken or there's not a variable that's not working. A negative control is the opposite. Negative control is an experimental control that does not give a response to the test. Again, the scientist determines how they're going to do this. It gives a negative result, no response, and it ensures that because there's no response to the test, you know that there is no external influence that is causing a positive response that you might accidentally say is from your experiment or from your independent variable, when in reality it is just a natural external factor that you did not take into consideration. Let's look at an example of some control groups. So in this example up here, this is our experimental group. These two are both control groups. So in this experiment, amylase, which is an enzyme that breaks down starch, it's getting added to starch. And so you can see here and here, they're put together. And what's going to happen is there's going to be a reaction, and eventually this amylase will kind of eat all of the starch and you will see a color change. But we need to know, as we're doing this experiment, is your amylase pure and is your starch pure? And is the amylase actually breaking down the starch to cause this reaction or cause this result? Or is there something else going on? So your control group here, you have amylase with distilled water. And this is a negative control group, right? Because I am not adding starch. Without starch, amylase should not do anything. I should see a different result here, or no result compared to my experimental group. This is also a negative control. We're adding the starch solution to water. And I know that this is a negative control because nothing should happen. Starch doesn't react with water. So I should not see anything happen here. If I saw something happen here or here, then I would have to redesign my experiment because this result would not be accurate because something else is going on with either the amylase or the starch based on the controls. Another way that you will see a control group is here. So we said before that the control group might be something that's not getting the treatment. Well, in this example, when you're looking at some more of an observational, you look under normal conditions. So normal would be the control group, which is normally 8.2. So when you move away from the normal, those are your experimental groups. Sample sizes of your experiment are really important to think about as you're designing. The bigger sample size you have, the better. And it will all make sense when we start doing statistics about why that is. So for example, if you are doing an experiment with time, you want to have a range of time. You want to have if you're doing it with generations, you want to have more than one generation so you can see a trend. If you're taking a sample of a population, right, because we can't test, if we're doing humans, you can't test every human on Earth. You're taking a sample of the population. So you want to take a sample that is as representative of the whole population as possible. And so in that case, the bigger, the better. We usually try to get 30 is a good sample size. And also, when you are doing an experiment, do you need there to be more than one trial, right? Because if you only do one trial, you might get a weird result that is not representative of what would happen if you kept doing it over and over again. Collecting data is the most crucial part of any experiment because this is what you are going to base your conclusions on. It is important in every experiment that you have organized data tables that are labeled correctly with the units. You do not just want to write numbers everywhere because you might not know what they mean. When you are collecting data, you need to be precise and as accurate as possible. You do not want to guess. You do not want to estimate. You want it to be as perfect as you possibly can make it. An important aspect to this is think about how you are going to measure before you start the experiment. Have a plan. Know what you're doing and how you're going to do it the same every single time. In the next video, we will be talking about what to do with all of these numbers collected during our experiment. How do we analyze them to come to an appropriate conclusion? See you then.